Thanks, Jack. I'm Andrew Lackner, and I'll start this off. And I think, as was said, with the topic of HIV/AIDS, you really the focus is on non-human primates, and to some extent, the One Health and non-human primates, there may be a bit of a historical advantage because the whole concept of the National Primate Research Center program goes back to really the late 40s, early 50s, and was driven by what was then the National Heart Institute, in a desire from particularly the leadership of NIH and a number of pioneering MD investigators to come up with a model of cardiovascular disease that would be more amenable than trying to use humans. So there's that history there, and there's a history of really multidisciplinary, transdisciplinary research that I basically have spent my entire career in. So, Jim and I have been collaborating on specific projects now for over a decade, and we each also have our own research projects, our own independent careers before we started working together. But I'll certainly tell you that the idea of the prior speaker about getting rid of the box, I mean, it is indeed convenient to stay in your own little world, but the most fun is when you're working with people who bring different perspectives. and that's something that I've experienced over and over in my career and has really made it a great adventure. So we're principal investigators on our own projects. We have several shared NIH projects, and we'll be talking a little bit about that. But in addition, Jim and I each actually have institutional roles. Myself as a director of a National Primate Research Center, and Jim as a director of a Center for AIDS Research, where we foster interactions amongst our faculty to combine the very synergistic expertise of a Center for AIDS Research and a National Primate Research Center. So because we have these sort of multiple roles, I'm going to spend some time talking about the National Primate Research Center, the Center for AIDS Research, our individual projects, and then we'll have a conclusion. So I'll give an overview, talk about the non-human primate model of AIDS, the research team, then I'll turn it over to Jim, who will talk more specifically about the science that we've been engaged with for about the last decade. He'll talk about the CFAR, and then I'll switch back and talk about some of the collaborative pilot projects, and then we'll have conclusions and discussion. So with regard to a primate center, the mission is a pretty simple one. It's to improve human and animal health through basic and applied biomedical research. That mission has several parts. The first is sort of the traditional one of a research center conducting basic and applied biomedical research on human health problems that require the use of non-human primates. And that word require is an important one. Relatively speaking, there are a small number of non-human primates that are available for biomedical research, so you basically have to focus on things where the work in non-human primates is the only way you're going to get the answer. That's where they provide the most value and the greatest impact. Because of the funding structure of a National Primate Research Center, we also investigate non-human primate biology and diseases, and that's actually where a whole range of models came from. That's where the non-human primate model of AIDS came from, and many others. And then we serve as a regional national resource and center of excellence for biomedical research using non-human primates, and that was what I was alluding to with the history. That's the reason the primate center programs exist, because individuals with a great deal of foresight in the 40s recognized that non-human primates were going to be really critical for biomedical research, but that the resources required, both the physical resources and the intellectual resources, were such that you couldn't duplicate them at every medical school in the country. And then, of course, we have a very significant educational training component with regard to graduate students, veterinarians, postdoctoral fellows, and so on. At the Tulane Center is part of health sciences at Tulane, and I'd point out that our roughly 40 faculty have appointments in 7 different departments in 3 different schools. So by design, we are highly multidisciplinary, highly integrated, with other components of the institution, including the School of Medicine, School of Public Health and Tropical Medicine, and Science and Engineering. So in addition to DVMs, MDs, and PhDs, we also have some biomedical engineers and others that are involved in the research at various points. We're one of 7 NIH-supported National Primate Research Centers. The primary focus of work at the Tulane Center is on infectious diseases, although we do other things. And one of the ways that we continue to sort of foster the engagement of multiple disciplines is through a pilot research program that enables people and is actually focused on people who have perhaps not done work with non-human primates before, also encourages junior folks. And I'd mentioned that one of the, what is now a collaborative R01 that Jim and I have actually started as a pilot project a number of years ago. And then, to really make things work, the center has a whole range of resources that can be plugged into with integrated clinical and laboratory support for non-human primate studies, and a full-time staff of veterinarians, pathologists, and technical folks who have a great deal of experience in a wide range of studies, including those that require BSL-2 and BSL-3 facilities. This is an overly long list of some of the clinical and laboratory capabilities. We do a large amount of minimally invasive techniques so that you can get repeated samples. As I mentioned, relative to most species, there are not large numbers of non-human primates, so being able to get multiple samples from animals over time becomes really critical. And the clinical imaging aspects go along with that as well. And then we've also recently begun doing biotelemetry for real-time continuous remote physiologic monitoring, which actually has been quite enlightening. You'd be surprised what you can see when you're measuring temperature not once a day, but 100 times a minute. And then there are a whole range of cores that people can plug into, anatomic and clinical pathology, confocal microscopy, and so on, that enable a whole range of investigators to do various research projects. And then you have things like infectious disease aerobiology, which are quite unique, which are looking at aerosol delivery of drugs, of vaccines, also of agents, and then also is involved in looking at how various agents survive in aerosols and what makes them survive or not. The non-human primate model of AIDS is one that has been very widely used and is extremely multidisciplinary in many different ways, and actually also incorporates some of the idea about the environment as well. So there's a fair amount of work on the origins of HIV and cross-species transmission and some of the environmental and cultural changes which have occurred in Africa over the last 50 to 100 years. Some points here that will become critical for some of what Jim is going to talk about is that the non-human primate model has demonstrated that a vaccine for AIDS is even feasible. The first pathogenic molecular clone of a lentivirus was developed in this model. And because of that, you can really very carefully dissect host and viral determinants of pathogenesis, which are almost impossible to do any other way. And that the model serves as a testing ground for vaccine and microbicide concepts before products enter human testing. So you'll note that I have the word models there, and indeed, as this model or models have developed, I refer to animal models kind of go through some stages, and sort of the first stage, which some of the questions that were asked of the prior speaker, is the Me Too phase. That's where you have to show that what you're showing in your model actually reflects what happens in humans. But when a model passes that point, it then starts to predict things that are going to be happening in humans. And in many ways, that's a wonderful thing to happen. It also ends up being rather challenging, because if your model is actually any good, what you end up doing is showing things that people don't know and that they don't think happen in humans. And I've seen that happen multiple times, where the models get out in front, which is where they should be, in terms of showing aspects of disease that are unknown in the human human condition which you're trying to model. And that some people have said that this model, and these are MDs, may be the best animal model that is in existence. So this model has actually become multiple models, and it's used to look at pathogenesis and correlates of control, of protection, and of disease. It's used to test various hypotheses, drugs, and vaccines. And of course, the question is which model to use. And what's happened over time is with some of the molecular techniques, and as we know more, you have a whole range of potential combinations. Same virus, different monkeys, and of course, people being people, you also have some fun things like it's the same name but a different virus, or a different name but it's the same virus. And then there also are models with it's the same, could be the same virus but a different stage in the life cycle. So there's pediatric models. There's models of aging in AIDS that have also been developed. And of course, which model to use, it basically depends on the question. And you want to use the one that's best suited to the experimental question of interest. So with respect to the research team that Jim and I have, this is actually not a complete list. This is sort of the core group. And as you can see, it includes veterinarians, MDs, PhDs, dual degree folks, and a variety of postdocs and students. And I would just tell you in terms of functioning, it's very highly collaborative and interactive. It's very much problem-based, hypothesis-driven, and multidisciplinary. And it is those times when you're multiple people are looking at a particular problem in different ways that are the most exciting, the most fun, and where the insights come from that lead to breakthroughs. And the focus is very much on the skills, abilities, and capabilities that people bring to answer the question. And quite frankly, the labels are not relevant. We never talk about them. It's always focused on the science. It doesn't even matter, clinical versus basic, applied scientists. Those are labels which people certainly use, and they can be useful, but in some ways I think they can also get in the way. And so we always try and focus on what's the critical scientific question that we're going after, and who has the expertise to get at that question. And if it's not somebody in the room, we go find somebody. So now I'll turn it over to Jim.
Okay, thanks, Andrew. So the story that Andrew and I are going to tell you is one in which the vast infrastructure of the Primate Center network, and specifically the Tulane Primate Center, was able to be applied to a scientific question that I approached Andrew with as an investigator several years ago, and we're going to talk a bit about that. But the bigger picture in the context of this meeting is how that has not only been important for what you're all thinking about today and tomorrow, what we're thinking about, in bringing a team together, a complex team to address the questions that were posed, but also to then, as center director to center director, how we took lessons from what we learned as individuals and then began to applying it in a larger context, namely that of the CFARs and how they can work with primate centers. So first of all, I am an MD, not a PhD, and I'm actually really am an oncologist. So I just finished 2 weeks in the hospital of the University of Pennsylvania bone marrow stem cell transplant oncology unit. So that is really what I do. But I have had a lab for 30 years that's focused on many themes of HIV research. And so to tell you a little bit about the science and the question that I approached Andrew with several years ago, I want to give you a brief intro into some of the key themes about AIDS, HIV pathogenesis, and then we'll transition into the specific project, or at least one of them that Andrew and I are pursuing. So whether you're talking about a human with HIV infection or an Asian macaque with a pathogenic SIV, the themes that play out are that very early on, there's a rapid increase of virus when it gains access to the system, either intravenously or through mucosal barrier, some variable decline in virus when immune responses kick in, and then some set point that reflects an active, ongoing proliferation of virus that in pathogenic models leads to the progressive and typically irreversible destruction of CD4 cells in the absence of therapy. So what we've learned about these models in humans and in macaques is the importance of the early events. So we know that this process is driven by viruses that all have in common the ability to lock onto the CD4 molecule, and then with conformational changes to engage a chemokine receptor, usually CCR5. So that's how they get in, that mediates their targeting. In the context of how this plays out in a mucosal barrier, we've learned that there are actually a very limited number of viruses that get across. But as they engage CD4 cells, there are so-called broadcasting events that recruit other CD4 cells to these surfaces that amplify that local infection and take it to the rampaging systemic infection that shortly follows. And in work that Andrew really did in the non-human primate model several years ago, before it was appreciated that in fact this happens in humans, we've learned that that much of the amplification that takes place in the early phases is a result of HIV infecting CD4 cells in the gut, where there's massive replication, massive depletion, and what we now know is as the destruction of epithelial barriers that lead to microbial products getting across, systemic immune activation, and ultimately through a complex set of events, creating the milieu in which ongoing viral replication is coupled with ongoing chronic immune activation. And it really takes the two to go hand in hand to take individuals or monkeys to AIDS. So the early events, CD4 tropism, mucosal targeting, immune activation being fundamental themes. And of course, once the virus gains its foothold, it replicates very rapidly and is then able to mutate, which enables it to escape host immune responses that kick in. So what we've appreciated more and more over the last few years are the importance of location, where this virus goes, what it targets first, but also the timing, how quickly the early events play out, and the order in which they play out. And so the scientific question that Andrew and I have been focusing on for the last several years have really framed in the following question. Can rational perturbations in HIV and simian immunodeficiency viruses, pathogenesis, reveal vulnerabilities that can be exploited for vaccine and therapeutics? So if there's an ordered orchestration and timing of what this virus does and where it does it, how can we mess with this to learn things about the host-virus relationship? And there are really two models that we've worked with. I'm going to tell you about one of them, but they rely on the pathogenic molecular clone SIV MAC239, which in Asian macaques typically leads to AIDS. So in one model that I'm not going to be telling you about, we were able to create with a student, PhD student at my lab, Adrian Swanstrom, a virus termed IMAC that is dispensed with its need for CD4. So this virus will use CCR5, but it lacks CD4 tropism because we bred it in the lab to do that. So this virus will get into animals, replicate quite well, but it loses the targeting of CD4 cells. So that is a very interesting story. But the one I'm going to be telling you about is a virus that through a small change that I'm going to describe for you has lost the ability to target gut CD4 cells, which I told you were among the most important early events that play out. So both of these projects have led to R01s and a fruitful and very enjoyable collaboration. So I'm going to be telling you about a virus termed delta GY. And in a nutshell, what delta GY is, it's a model in which a 2 amino acid deletion, only 2 amino acids, in this pathogenic molecular clone renders this SIV highly susceptible to host control while retaining a wild type level of replication. I'll show you that in a second. But importantly, once control of this delta GY virus occurs, animals go forward to be not only healthy, but to become broadly protected from pathogenic challenge viruses. So controlling viruses and what we need to protect ourselves from pathogenic challenges are fundamental to the key questions that HIV researchers are asking all over the world. So we won't have time to really go through the details. Jack is going to be cracking the whip if I violate my time here, but suffice it to say that here is the diagram of the part of the envelope glycoprotein with the membrane spanning domain, the cytoplasmic tail that starts here and actually goes on for about 150 amino acids. And what we recognized early on is that there is a highly conserved trafficking signal in the cytoplasmic tail, universally present in all HIVs and SIVs, and it mediates a number of functions that I've just listed here. Importantly, it's a very powerful endocytosis signal serving to downregulate envelope from the cell surface, pulling envelope that's not incorporated into virions into clathrin-coated pits. So why is it so conserved? What does it do? It actually has very little effect in vitro. And so what our lab did in collaboration with Mark Marsh and University College London several years ago was to explore viruses in which there were mutations. And with MAC239, we destroyed this signal by a 6 nucleotide deletion, eliminating the G and the Y, creating the delta GY virus. So it shows a diagram of what this virus is like. Again, it is identical to this highly pathogenic MAC239 in every way except for 2 amino acids. And so what delta GY did in rhesus macaques was interesting in that it went to a very high peak, so delta GY shown in blue, a high peak early on. There was a greater degree of host control, but ultimately animals ultimately succumbed to AIDS. There were things that we learned about this model, but the one that I want to tell you about that was rather astonishing was that when we looked at one of the non-human primate species that Andrew referred to, when he said there's a lot of decisions you make. If you look at the pigtail macaque, which is well known in the field for being highly susceptible to virulent outcomes, this delta GY virus had a very different outcome. You see it replicates early as well as MAC239, but with the onset of host immune responses in 19 of 21 animals, they basically became elite controllers. So the paradox is that this small change in the most pathogenic or virulent species led to a virus that was better controlled, but in the species where disease seems to play out more powerfully. So what's going on? And it's at this point that we really needed the power of the Tulane Primate Center, as Andrew has told you. And a lot of work went on to showing a number of themes that played out in the context of control. First of all, there's no immune activation. There's no disease progression. Once this virus is controlled, these animals are perfectly fine. Rather remarkably, there was a sparing of gut CD4 cells. CD4 cells in other anatomic sites were rapidly infected, but not in the gut. There was no macrophage infection. And we learned that the fact this virus persisted, there were CD8 cells that were controlling it, but was remarkable that to Andrew, who really described the effect of HIVs and SIVs on the gut, the CD4 cells in the gut not only were present, but mounted really unprecedented polyfunctional, very powerful antiviral CD4 cell responses. So a very, very different outcome. And just to kind of show you pictures, I mean, to be able to say things like this, you have to have in situ hybridization, you have to do biopsies, you do multi-label confocal microscopy, flow cytometry, and it's all done at Tulane because of the vast number of PhDs, VMDs who are able to run these instruments and give us this kind of information. So importantly, though, animals that control delta GY were being healthy and being able to live for years, when challenged with a number of viruses, had this remarkable outcome in that just for a single animal, I'm showing you that giving them serial intrarectal challenges of a heterologous virus, a genetically different virus from MAC239, basically now we're looking at E660 virus would be in red, delta GY in blue. You can see the controlled delta GY here. We got no infection here. If you give it IV, we get no infection. And if we counter with a highly pathogenic SHIV, an SIV that contains an HIV envelope, we get no infection. There were controls for all of these challenges, and the naive animals all rapidly succumbed with high viral loads and AIDS. So it, of course, raises the question, what is this control all about? Is it antibodies? Is it neutralizing or non-neutralizing functions of antibodies? Is it cellular responses, those hot polyfunctional CD4 responses? But these are questions that are absolutely mainstream to the field, and this is much better than any of the published models of so-called attenuated viruses. So we think it's a very powerful model to carry forward. The last thing I'll tell you in terms of the science is that we've collaborated with Michael Cates at the University of Washington to look at transcriptomic profiling of these animals. And to just lay out in a minute what we're able to see, if we look at delta GY in RNA transcripts from the blood 9 days post-infection, delta GY here, MAC239 here, genes that are going up are in red, genes that are going down are in blue. At 9 days, delta GY and MAC239 are identical. They're all taking off. They're establishing identical levels of viremia. But you can see there are remarkable differences that are playing out. Myeloid markers going very high with MAC239 and not present here. Interferon responses occurring in both, but ultimately cooling off later on in the context of delta GY infection. And small nucleolar RNAs going to very high levels in delta GY and being rather neutral in MAC239. So it takes a lab like the Cates lab to sort out what's going on, and we're actively in the process of looking for this, for the signatures of what's different. So the bottom line to this interesting scientific story is that it takes a village. You know, a 2 amino acid change, producing this kind of change and describing it, and the things that I've just told you, has required an input from a lot of people. So Tulane, and then my team at the University of Pennsylvania, but much more than that. The Jeff Lifson and Mike Piatak at Frederick, at NIAID and VRC, the Wisconsin Primate Research Center. I think David O'Connor is out there somewhere. He's been extremely helpful in this. Duke, University of Washington, Mark Marsh, University College London. And I want to point out the critical role that the Division of AIDS has played with us in paying attention to this model and giving us support in a number of ways. And I also want to mention in my lab, Samra Elser, a vet PhD student. We have a vet school at the University of Pennsylvania. She is a combined degree student who heard about this project, came to me, said, I really want to work on this. And she's spent some time down at Tulane with Andrew, a terrific training opportunity for her. It was a debate of whether to use this picture or the one of Samra with her golden retriever, named Delta, by the way, not Delta GY, just Delta, who happens to be having an osteosarcoma now with a meningioma, Carolyn. So I don't know about meningioma, but she's absolutely right about the challenges of having golden retrievers. All right, so in the last few minutes, I want to go from the science back out. As Andrew and I were enjoying this collaboration, we began to ask, what could we do as center directors to take the benefit of what we've experienced together and make it more available? What have we learned as individuals that we could then roll out? So at this point, Andrew told you about the National Primate Research Centers. I'm going to be telling you about CFARs, and then we were able to take our very positive experience forward in an inter-institutional sense. So we're going to talk about CFARs and then the nature of how we brought CFARs, the CFAR network, and the primate centers together. So what are CFARs? So Centers for AIDS Research. In 2014, this is the map of where they are. Centers in green are so-called developmental centers, but there are roughly about 19 of these. I'm at the University of Pennsylvania, where our center includes all of the University of Pennsylvania, including its vet school, as well as Children's Hospital and Wistar Institute. And the mission of the centers of this NIH program for all of them has basically been with research, education, international components, community outreach. It's basically how can you do more together, do things in an interdisciplinary way, show that you can do more together than you were doing as individuals, get out of your silos the whole bit. We don't need to go through this, but that's what NIH had in mind in doing this. And the CFAR network has been an extremely collaborative group that I think has really worked not only on their campuses to do these kinds of things, but between their institutions. There are many examples of that. So the one that of course, we're here to pose to you is how we began to think about what the CFARs could do with the National Primate Research Centers. And you can see there are 7 of these, and there are various locations, not all of them being at where CFARs are, but the science and the importance of this model that Andrew introduced to you and that I gave you at least one example of, really speaks to the need for the resources of primate centers and the questions posed by scientists of all kinds at CFARs to bring these together. And so our CFAR, we at At Penn, we've had it for 4 cycles. We'll have had it up to 20 years by 2019. And as I said, it does include our vet school. And our CFAR, like many of them, sort of looks like you've got research areas, and I won't go through them in the interest of time. But importantly, all CFARs with their funding from NIH create cores designed not to duplicate things that are already on campus, but what does your community of HIV, SIV researchers need to make their research go forward. We all have a pilot grants program, and then recently we have scientific working groups that are focused areas of emphasis that we keep for short periods of times and then cycle them out as other ones come in. So this red star is to refer to our core at the Penn CFAR, which is a remote core. And what we did was really incorporate the Tulane Primate Center as one of the cores. So we have our non-human primate core based in Tulane. It's no problem at all to communicate with Andrew and that team of people that he introduced you to. And so we have really brought Tulane into the context of our CFAR infrastructure. And Andrew's going to tell you a bit about what that core does, but the last thing I will tell you is that we felt very strongly that what we needed to do at Penn in Philadelphia in the context of this new relationship that we crafted in 2004, I think, Andrew, is when we kicked this off, was to really make the Tulane investigators feel like they were part of Penn, and for them in turn to make us feel that we're very much a part of what's happening at Tulane. And so as an example of this, we have every week as a series, the HIV Grand Round series, live broadcasting. It goes to over 100 domestic sites, 15 to 35 countries, all the lecture of archives. So our archive, it's a wonderful resource. Here's Andrew being invited to Penn, and there have been several investigators from Tulane coming to speak at this series and others. But again, just driving home the point that Tulane and Penn are very much integrated in the context of the science. So with that, I'm going to turn things over to Andrew, who will tell you what this core does, and then we'll open this for questions.
Thank you, Jim. So, yeah, the collaboration between the CFAR and the Primate Center has really been extraordinary and highly synergistic and bi-directional. The core actually helps make the non-human primate model very accessible to faculty at Penn. We provide a lot of advice in terms of non-human primate studies and mentoring and encouraging, particularly junior investigators and involved in training, as was mentioned with SAMRA. We host investigators and their students who come for variable periods of time, depending on their projects and time. Take care of all the fun regulatory stuff that's involved, and then provide essentially all the infrastructure for doing the non-human primates research. And then in addition to the pilot program that's part of the Tulane Center, there's a pilot program funded from the CFAR. We basically fund a couple of projects a year, about 8 animals per year. We've had 21 pilots supported since 2004. This has led to multiple R01s, program projects, many publications, a lot of mentoring opportunities for Penn students to visit the Primate Center, and we've had jointly sponsored symposia on the non-human primate model of AIDS that have been very exciting and fun. And then the Primate Center faculty also go to Penn. At the bottom, these are just a couple of very recent grants that resulted from pilot projects. They were both funded within the last year. One with Dennis Colson that is actually looking at the neuro-AIDS model in non-human primates and how HIV, SIV affect the CNS. And then a second project with Michael Kinkro and Gene Schultz. And this one is of note because Gene is a junior investigator, and it was the pilot project and getting that off the ground led to this. A big part of the push was on helping out some of the junior investigators, and that's looking at BLISS and its ability to change how antibodies evolve and perhaps provide more broader reactivity of the immune response. So the two pilot projects, pilot programs, one that's funded from the Primate Center, one that's funded from the CFAR, are really very major initiatives that help foster these sorts of interactions and have been extremely beneficial to both groups. So in terms of conclusions, I think it's probably clear to everybody that biomedical research is increasingly complex and multidisciplinary and requires collaborations among individuals with varied degrees and diverse skills. If you look at a paper now in a reasonably high-profile journal, and you look at one 10 years ago, there's almost no. The differences are so vast in terms of the amount of data that's required and the variety of different techniques and technologies and approaches that are required. This is basically the norm now, and I think it really drives a need for One Health of these highly multidisciplinary teams and quote unquote team science to really be able to make progress. And I think from our perspective, these highly collaborative multidisciplinary networks, of which the CFARs and the NPRCs are one example, we think are positioned to develop very innovative approaches to complex problems, to address research gaps, and to translate research to relevant areas for humans, that they provide an infrastructure and emerging technologies and animal models that can be leveraged to create new scientific opportunities for investigation, and perhaps most importantly from an educational standpoint, provide unique multidisciplinary training opportunities for veterinarians and other biomedical scientists. And with that, I will end. So I think Jim and I.
Andrew, great, and Jim, great overview. Nice collaborations. Congratulations on that. So in terms of thinking about maybe what is missing in our training for veterinarians in research, Andrew maybe, or Jim, either one, you have a great environment there for getting them directly involved in One Health projects and primates. But is there things missing that we could do in our communities or in collaboration with NIH that would enhance training to get more veterinarians as primary investigators in research?
I think being exposed and being involved in these sort of larger multidisciplinary groups is really important. I think it provides perspectives particularly for veterinarians that of what's out there and what's possible. And I think that and you have to have the sort of the role models as well. But I think there is to some degree with respect to veterinarians, some trepidation about working at a medical school, working with MDs, and that somehow you're different. And I think we just got to get over that. Many of the labels we use are historical, and things are moving so fast and changing so fast that I think in many ways they hold us back. You have to have some way of organizing universities and schools and so on, but research, the research questions don't care. And so I think the more you can do to get people involved in those kinds of groups that involve multiple disciplines, the better off you'll be. That particularly for veterinarians, the less trepidation there will be about walking into institutions like that and getting involved. And it's, from my perspective, it's extraordinarily rewarding and exciting, to work in that sort of a team environment, because people are always bringing things from new angles that change your perception of whatever the problem is in ways that are often very unexpected.
One quick thing. So one of the things where we benefit from clearly is at Penn having our vet school right on the campus. So 2 blocks here in the vet school. It's very integrated, our combined degree program. It's a joint review process where lots of interactions with basic science faculty during the interview process of the combined vets. But that said, when I told Samara Elser that I was coming here to give this talk, I said, I'm doing this thing with One Health. What is that? And she says, oh my gosh. You've never heard of One Health. So then I had to admit that I was not well educated in One Health. I'm going to be much better after these next couple of days. But yes, we can, even though Penn Vet School is at our campus, I didn't know what One Health is all about. Maybe this is more about me than our campus, but we can always be better at communicating.
You were doing it.
So this may be the corollary to the question that was just asked. We find that the veterinary students, now the students, are very zealous about One Health, and they know all about it, but the medical students seem not so well informed. What are some of the corollary techniques we might use to reach the medical students, to have them understand the value?
Yes, you've nailed an important point. I think it needs to be integrated into the curriculum. And so, medical school is such a good time to get to them. And by the time they're residents, they're too busy and they're locking into their ways of thinking. So I think the concept of One Health and what we're talking about today needs to be brought into the curriculum. And there are many, many places where it could be, as is clear. So, but as Andrew's saying, sometimes we're talking about One Health, but we don't know it. So framing it differently. But I think the answer to the question has got to be brought in early to the medical education, or it's not going to happen later on.
The important thing is that people do it. I guess my perspective on that is that, I think things have changed over the last decade or so. I mean, I think I have my concerns about study sections in general that have nothing to do with animal models or veterinary medicine. And I think that's actually, in many ways, is a separate problem. I mean, I've not experienced really any situations where there was, where the problem was that there weren't veterinarians on the committee. I mean, I have had, experienced multiple times where you had people on the committee who were reviewing things that they didn't know what they were talking about. So I'm not sure that that's a DVM, MD, PhD thing. I think that's a more general issue of making, of getting the right people to review, and as often happens, the people who know the most are the ones who are the busiest and have the hardest time being involved in some of those committees.
We're going to have to stop. Please feel free to catch the two of them when you can in the break. So we now have a break from 10 to 10:15. We will start promptly at 10:15 to stay on time. Thanks a lot.